So what is purification? Purification of enzyme is a series of processes in which we isolate one or few enzymes from complex mixture, usually cells, tissues or whole organisms. Enzymes are purified because of their role in different industries like agriculture industries, cosmetic industries and pharmaceutical industries. The enzyme which is found in the cells may be intracellular or extracellular in nature. Sources of enzymes. So enzyme occurs in all living organisms and catalyze biochemical reactions necessary to support life. Microbial are the best source as compared to plants and animal sources because microbial sources are generally cheaper to produce. Their enzyme contents are more predictable and controllable. Regular supply due to absence of seasonal fluctuation and rapid growth of organisms, microorganisms on inexpensive media. Microbial enzymes are also more stable as compared to plant and animals. So microbes, different type of microbes are used as a source for enzyme production. Some are the example here. Escherichia coli enzyme which produce amylase, Bacillus species, Pseudomonas and Clostridium are the sources of proteases enzyme. Candida species and Aspergillus species are the sources of lipase enzyme, and Aspergillus niger is a source for catalase. So after the selection of source, and if the enzyme is intracellular in nature, the cell lysis is an important step in during protein purification or isolation of our protein interest. Cell lysis can be done by using different technique using detergent lysis, osmolysis, freeze-thaw cycle, enzyme lysis, ultracentrifugation and homogenization. Different type of enzyme are used for the lysis of cells, including lysozyme for the lysis of bacterial cells, is for fungal cell lysis, and cellulose for plant cell lysis. After cell lysis, when the crude is removed from the cells, the protein get precipitated by using salts called ammonium sulfate. Then we go for dialysis and different type of column chromatography, including size exclusion chromatography, ion exchange chromatography and affinity chromatography. So in case of ammonium sulfate precipitation, we use ammonium sulfate to precipitate protein. Ammonium sulfate is basically used because it is more convenient, effective, high solubility, lack of toxicity and stabilize the effect of some enzymes. After ammonium sulfate precipitation, the most important step is dialysis to remove the ammonium sulfate, because ammonium sulfate when ammonium sulfate is occur with protein in long period of time, it can denature the protein. So ammonium sulfate is removed by process of dialysis. So in dialysis, removal of small unwanted compounds from macromolecules in the solution by selective passive diffusion through semi-permeable membrane. So in dialysis, there is a dialysis bag in which we add the protein, and with ammonium sulfate is out from the protein because of their size. After dialysis, protein purification is done by column chromatography. At present, there are numbers of chromatography methods that can be utilized to purify protein sample. In case of ion exchange chromatography, in which different type of proteins are separated based on their net charge, as such column can be either be prepared to facilitate both ion exchange and anion exchange and cation exchange chromatography. Other technique used for protein purification is gel filtration chromatography, size exclusion chromatography and affinity chromatography. Here this is the diagram showing the ion exchange chromatography, in which mixture of protein are added in a column which is packed with a resin which have some charge. So if suppose if we want to a protein which have negative charge, so opposite charge region was packed in the column. Remaining protein are elute out from the column. gäller det